This
community
conference will now be recorded.
has been started to look at the post-translational modifications. Yeah. So, and why they are very important? Because, well, there are many many more importance, but basically it's signal transduction, the enzyme activations, molecular recognition. So all these processes are happening at the level of, let's say, pathogen cell recognition. And if you want to engineer something which is an enzyme that has an inherently an active capacity, so you will have to address these problems, right? So here I can, I mean, this is our favorite protein in the lab. Basically, we have been studying the spike protein, which is pretty much is very large protein. We are talking about 3,000 amino acids. So it has well-known two states. One is the closed, which is basically the inactive, and the open one, where one of the, let's say, 200 amino acids, that is the one that is called the RBD, binds the cells. Well, it recognizes the cell receptor, and then it gets into the cells, right? And so this transition has been, let's say, barely characterized for the wild type form. But of course, we know that the spike protein has also variants. And if we use all the computational power that is available nowadays, it will be impossible to do all a systematic study of all different variants, right? But we know that if we plot, for instance, free energy, which is, this is, let's say, the distance between the RBD and another protein, and the angled transition, for instance, from this coordinate to that coordinate when it closes, you will see that all atom simulations are pretty much sampling. I mean, these are really data from very well-known papers. So all atom simulation is only able to sample around the minima. So in order to see the transition from the open to the close, you need to use some enhanced computational methods, which is very much restricted, even in Poland nowadays. Okay, so this is just an example. I will try to describe how we get to this point where you can observe the transition with much less computational power between the open and the closed state for the wild type. Okay, so the idea is that we could observe those processes with much more well, with the same accuracy in terms of the chemistry that describes those processes, as well as the inherent ability to see transitions between open and close. Okay. So once again, so I need to explain a little bit on the methods that we have developed. So everything is rooted on the idea of the native context in proteins. So it's a very robust idea that can be also exaggerated, let's say, and move into the different biomolecular systems, like even in the case of nucleic acids. Mm-hmm. So it has been a long time ago when I started this collaboration with Professor Marek Cieplak at the Institute of Physics, where he had a very beautiful model called the Go-like model for proteins. So his model was able to capture the intrinsic conformational state of a protein, which is just the green backbone, and removing all the different amino acid side chain. So the model was able to capture the equilibrium conformation of the protein based on what we call native contact. The native contact, where basically you map native contact based on the interaction between side chain. Two polar or two charged amino acids, they will attract to each other and they will be closer. So this is called structure-based modeling. So it's similar to the, let's say, the overlaps between grapes. And once you find an overlap, you will construct a potential that will be basically retaining the position of the two C alpha backbones, right? So then later came another method, which is the RCSU, that was also developed under his supervision. And basically it introduced some chemistry on the determination of the native contacts. So you can imagine that some contacts, they can be very close to each other, like the blue one. I mean, and then you have another contact, which depending on the position on the backbone of the protein, they will be shorter in distance, right? So they will be parameterized with the Lennard-Jones potential that is basically here, and there will be two parameters. One will be the, let's say, the sigma of the Lennard-Jones potential that will be connected to the distance between the two positions of the C alpha atoms. And then you will have an epsilon of the depth of the Lennard-Jones potential. So this model was able to capture, let's say, the dynamics of backbone of the proteins, and in this case, you could also study some processes like unfolding and refolding of the protein at different temperatures. So the model was sensitive to the temperature, was sensitive to the mechanical forces, but it was, of course, simple because it neglects the presence of this amino acid change. The C alpha, well, here is just the model of the C alpha backbone. So you will have the backbone by NCCO, so nitrogen, carbon, carbon, and oxygen, and you will take one of the carbons. So it's called this one of the carbons, the C alpha carbon. So it's one carbon just after the CB atom, which is the one that leads to the side chain here. But it's just one carbon. It could be also the CB atom, but so by, let's say, in proteins, so most of the, let's say, the community always has chosen the C alpha as the, let's say, the degree of freedom that can take care of the, let's say, the topology of the protein. So that was the basic idea. Of course, this method has been, I mean, what we learned is that we can actually get this contact map. We made a server, and every protein has that has a structure. We can construct the contact map, and based on the contact map, we can construct what we call the goal-like potential. So which would be a Lennard-Jones set of interactions based on the native interactions. Yeah? So basically, this idea led us to the introduction of the GōMartini 3. So let's say in those days, there was a Martini. So Martini was pretty successful to not only to coarse-grain lipids from the atomistic to the coarse grain, and then study these beautiful curvatures on membranes. Also was pretty successful to study the equilibrium properties of proteins by introduction of the harmonic bonds. Proteins basically in Martini potentials, which are coarse-grain potential, they require information about the structure. But they chose the elastic network as the potential to retain the structure of the protein. Of course, this limited the study of conformational changes for that system. And then, of course, I mean, it has been a long time ago since they developed parameters for sugars and also for nucleic acids, such as the DNA, and as well in the same, or 1 year later, for the RNA. So in this, let's say, very robust force field, so is where we developed the GōMartini. So it's nothing very complicated. So we had on the other side a structure-based model that is exactly what denoted also a structure-based model. The elastic network was also a structure-based model. So we basically removed the elastic network and we reparameterized the interactions based on the Go potentials. So this basically led us to, let's say, a bunch of new applications. Of course, we validate in our original paper on different protein models, as well as some, let's say, enzymes that they were having some very complex active loop, as well as some nanomechanical experiments. But this was done for globular proteins, okay? So later, so people actually got very much encouraged to use the models, and then they study protein complexes in lipids, protein ligand interactions, as well as the nanomechanics on large protein assemblies. And all this was done within, let's say, the Martini 2. So this was the second generation of Martini 2 potentials. And then, of course, recently, I mean, 2020, it came what is called the Martini 3 potentials. And then, of course, we team up with the whole Martini community in order to parameterize our GōMartini into the new version of Martini 3. So that one led us to the new paper. So it was just recently published in Nature Communication. And there is a lot of more authors than the original paper. But basically, each of us had contributed to have new test cases, and we have validated in different systems such as IDPs, protein ligands, and also some condensates and stuff like that. Okay, so of course, the work has been enhanced by many others also. So the study of multi-basin transitions using also the GōMartini. So a new, let's say, OLIPS is a method that actually use Go potentials, but to retain the conformational state of the protein, so which much more chemistry in the determination of this context. And also the study of oligomers, which basically can enable the study of very long fibrils proteins. And recently also the switching GōMartini, that is a multi-state system. Well, all this basically has a potential to even increase even further. And one of them is the study of what we do here, therapeutics and some applications on let's say, different systems. So our idea had been for a while to use the GōMartini to study different conformational states on systems that are very large, I mean, compared to single proteins, right? So we, at least myself, I dream that we can study kilodaltons of proteins doing all these, not only, I mean, in the crystallographic state, but actually to see the conformational changes and also how the complex system, they inherently increase their capacity to hold forces and also to respond to thermal activation, right? So, and when you think about pathogens and all these filaments coming out from bacteria, so you could think about those proteins, what is happening underneath. And for that, so we started to optimize even further our method. So this has been done over the last year. So what we know currently is that we have the Martini 3 potentials for the bonded part. We have everything which is electrostatics. We have the Martini potentials that have been reparameterized in Martini 3. And then we have the native contacts that had to be optimized. Okay, so this is the original formulation. But of course, we need to optimize now again the sigma parameters as well as the epsilons, right? And for that, we have chosen a method that is very traditional, which is the molecular dynamics, all atom molecular dynamics. But actually, recently also it has been powered by different activities in the whole community, which is the creation of databases. So these days, people are trying to do, I mean, to take a little bit the state where you can use all atom simulations to create a very large database, not only to reuse the simulations, but also to take it to the level that machine learning can learn from these databases. One of them is the MD database. And also there is the ABC consortium that is for nucleic acids, in this particular case of DNA. And then, for instance, if you go to COVID MolSim, so basically you will find all atom trajectories from the last pandemic for the case of wild type that are very much useful in case you want to parameterize a new coarse-graining model. If you know this data or you create your data, then you can basically reparameterize in order to find the high frequency context. So what are the high frequency contacts are basically the most relevant interactions in the trajectory that remain at the length where the high where they have the high probability to stay. We choose about 70% of the trajectory. So those contacts exist during a molecular dynamics trajectory 70% of the time. We have developed with our PhD student, Gustavo Olivos, together with also Fernando Cofas, and a script that basically takes trajectories from any of these sources or your own trajectories, and you can actually filter what is most important contacts. Having those most important contacts, you can create a contact map and then provide it to Martini 3, right? So, and then we test it, and then of course we need to validate whether the new set of contacts is also relevant. Okay, so this is basically the main idea. And how we do that. So basically, for the case of, let's say, a protein complex, this is an RBD from the SARS-CoV-2, and this is a therapeutic nanobody that is called potent nanobody because it has a high affinity to the wild type. It was studied by all atom simulations, just the wild type. But basically, you need a structure, whether it comes from, let's say, a crystallographic method, or if you use AlphaFold, you need to start with a structure. Once you have the structure, you prepare it, you remove whatever is not clear to you. So, and then you pass to the Martini tree. So nowadays, what you need to do to use a GōMartini, basically you need to install Vermouth. So it's the only main tool at the moment. So once you have that one, and once you create a contact map, or even these days, Vermouth has been optimized to create a contact map on the fly. So you basically you can also skip this step that you go to our web server. So everything is very much embedded on the Martini level. So once you have the, let's say, Martini, so then you can use Martini basically in the way to, well, you need an epsilon, you need to, if for people who knows how to set up a Martini simulation, you can include water, lipids, sugars, different descriptions of proteins, anything that you could remove basically. And so what you need is the structure. So it will create a topology. So it will create a coarse-grained structure. You need to pass what we call the ESSP that takes care of the secondary structure of the protein. And then you name the force field that is a parameter. And then the flag of the model, which is -Go, takes care of everything that is needed for the protein. So you just activate this flag, and then let's say the master line of Martini 2, it will be able to create in one click the model. So you don't need to do anything else, basically. So once you have this, basically you can visualize, and then you will see the, let's say this is the same model in the coarse-grained Martini, and the new interactions that I highlight the Go interactions at the interface are basically necessary in order to describe the well behavior of the system, okay? So now let me go to the applications. So some of them are basically a little bit old, but they are the basis of the, let's say, of the GōMartini. So single molecular force spectroscopy has been used, in my group actually to validate our experiments. So in collaboration, of course, with experts from the FM community, where, for instance, for the case of the AC2 and RBD from the spike SARS-CoV-2. So basically, you could actually follow the kinetics of the interaction of these two proteins in order to quantify, let's say, the amplification of the forces and also the binding events. So you could learn a lot of this, but the limitation is at the level of the speed. Experiments are happening at much lower speeds compared to in silico experiments, right? So what is a protocol that is well known is stir molecular dynamics. So this is, for instance, for an all atom. So you have the same protein, then you have the RBD, and then you pull with a, let's say, with a spring constant and a dummy atom. But of course, what happens? So what happens when you use all atom simulations? You will have, counted number of times, because this experiment involves a lot of water, and this simulation becomes terribly expensive if you want to repeat this, even if you have 2 mutations, right, on the surface of the RBD. Even if you take care of the delta, you will repeat this. This is a hard simulation. It's much longer. You will have to repeat this terrible simulation for a lot of time, several times, just to get an average profile that looks pretty noisy like this one, right? So with forces that actually change by 200 piconewtons or even more. So the certainty that you gain to see the average profile will be limited. And if you want to try in another variant, it will be, let's say, computationally very expensive, right? So the idea was to replace this setup by the coarse-grained simulations, right? So how we did basically that. So we started to look at some with some experimental data, of course. We look at the CTL4 anticaling that was a therapeutic complex. And so this case is basically well known. So experimentally, what they found before us was actually that they wanted to optimize the anticaling, the anticaling protein by basically pulling by in this case, pulling from different residues. And this will be the cartoon of the secondary structure of the red protein. And you have the N and C terminus also. So they, depending on the position where you do the pulling experimentally, you will see a change on the mechanostability of the protein. Right? This is a protein complex. This is a you can see that I mean, if you take the first and the last one amino acid and you start pulling, you will see that they will have the same values, but the anisotropy of the protein complex will actually be highlighted that if you pull from the 60 position residue, right? So once you pull, you could see that the mechanostability of the protein complex is actually enhanced by twice. And this was what we knew from the experiment. So they asked us to check what is happening at the molecular level, because there are different pulling directions and also, I mean, different, let's say, not so easy to do with all atom simulation. So we started to parameterize the model with the GōMartini. So there was only one parameter in the model, that was the epsilon of the model. So we started with, let's say, 0. We construct the coarse-graining model. So the interactions between the interfaces were basically set to 0, so which basically lead us to what is the blue curve. That is basically the naked Martini potential. So this is pretty much what Martini sees in the absence of the GōMartini at the interface. And then we started to parameterize, since we had the experimental data. So we started to parameterize the epsilon of the amino acids at the interface until we recover some, let's say, agreement in the profile respect to the experimental data. So these are done at much higher speed, of course, because the experimental setup is still done like 5 orders of magnitude slower. But if you consider all atom simulations that at the time were done 10 to the 9 times faster, so we were actually 4 times more, 10 to the 4 more efficient than all atom simulation. And since our errors are actually in the scale of some piconewtons, we could actually compare the profiles, right? So if you have all atom simulations, the errors will be pretty large and all the profile will be hidden by the error, right? Okay, so then we pass to understand, because we have the molecular video, so we could actually trace those processes. For instance, the most interesting ones, let's say the one that you start pulling from the first residue and the one that has the highest mechanostability at the residue 60. And you see that actually the pathways at Fmax. So Fmax would be the moment when the forces are going to be the highest, and then the protein complex will separate, right? So this is the instant where proteins actually are going to be disrupted from the interface, so this protein will be just removed. And when you pull from the residue 60, so most of the contacts at the interface, they will get activated and they will be, let's say, aligned to the direction of the pulling, maximizing the strength at the interface. So this, you could see also some motion. So this is the PCA of the center of mass of the protein complex. If you pull from the residue 1, so the system pretty much will actually stay at the beginning in the initial position, but at Fmax it will be, so the protein actually change side of the interface, is shift, and then at Fmax it's removed from that position. While if you take, let's say, the residue 60, the center of mass of the protein complex stays around the same initial position. After Fmax, there is a little bit of vibration where the center of mass shifts, but not that much like in the other case. And then it stays, so it actually tries to change a bit, but it remains due to the context in some specific position, right? So since we have control also on the different pathways, so this we can repeat this several of times, we can gain some statistics on the process also. And so if you remember, this was the cartoon of the secondary structure of the protein. So each of the beta, let's say, segments of the protein are being colored, and each one has a set of native contacts in the model, right? So when you pull from the residue 1, so you see that the most affected strands in the protein are the, let's say, the purple one, the blue one, and you could see this this basically is happening like a peeling of the protein. So basically you start peeling this one from that one, and then until the moment happens in these 3 beta strands, and then the system separates. While if you pull from the 60, the number of contacts remained steady over the simulation, which actually highlight the robustness of the interface once you set up the pulling from the residue 60. So, so then later, of course, I mean, this was an interesting validation also to give us some entrance to, let's say, a competition in order to understand a little bit better the mechanostability on larger systems. And we started this project actually not long ago, but well, we did some pre-work trying to position the idea that mechanostability is also relevant in protein complexes, like in that study. And this was translated to the SARS-CoV-2. And considering that the spike protein is a very robust system, so we could imagine also that there is some signature or footprint of mechanostability. We show that this is present at the level of the RBP. Of course, this has to be extended, and this has been also done over the recent years. But we moved to let's say, to the case of therapeutic antibodies, right? So we try to reposition this idea. We ask the experimentalists why you cannot do nanomechanics of antibodies. No, because they bind too fast. And we cannot have the resolution to observe the process once they bind too fast and then pull it apart. And so this is perfect for simulations because we can control the conditions and we can position the antibodies exactly in the moment where they are going to have or suffer, let's say, some mechanical forces, right? So what we learned from the previous pandemic is basically that the emergence of different variants, some of them they remodel completely the interface of the SARS-CoV-2 spike protein, and they show also some level of neutralization, right? So antibodies, they were binding natural antibodies, our body created them, so they were binding to the wild type, but they were, I mean, the BA1 and the Omicron and the latest one, they were immune to our, let's say, own defenses. So then, I mean, engineering antibodies was one solution, right? Or boosting, let's say, our immune system with vaccines, right? But basically, so there were, let's say, spikes on the pandemic, and Delta was one of them, right? And also, we try to place the idea that mechanical forces are kind of important at the level of the viral entry. So, what you can see in this cover image is that basically you have the virus and you have the cell, you have the receptor, and there is nothing else. But that's not true, right? So there are, let's say, the glycocalyx is here. So there is a lot of shear forces acting on the level of the spike, the virus. This is the first, let's say, protection of our body. So there is, we can name the different mechanisms that our system moves in, and different systems are trying to avoid that pathogens reach our cells. So this picture in the end is not complete, right? So we can coarse grain all of that and think that there are mechanical forces happening here. And also the whole variant is not small, right? So it's also moving and also inducing some stress on the cells. So these tiny proteins, which are actually sticking out at the level of the surface, they must be mechanically stable as well, right? So well, llama is not only a very beautiful animal from my home country, but actually it's been used also to engineer antibodies, right? So in this study, so basically, so it was a study in the past by all-atom single-stir molecular dynamic simulation, let's say the mechanical response of a potent nanobody, right? That actually responds in the range of piconewtons, right? And this is the receptor from the spike protein. So the profile of force over displacement, it would look like in the case of all atom, it looks pretty much like this, yeah. And so there were few trajectories because once again, you need to make these simulations and you don't have enough computational power. So you run, let's say 20 runs or even less. And also there is a limitation that when you apply a method like all atom whole atom simulations, you need to restrain the position of the protein to avoid what is known by the unfolding of the protein, right? So the force field doesn't like to leave the equilibrium, and once this happens, so the response of the whole complex is not exact because it has not been parameterized for that. While in our case, so basically we did hundreds of coarse-grained steel molecular dynamics simulation for the same protein complex, and then you could see the distribution of the forces around these maximum forces. And okay, so once you have this, then you wonder, so what is happening? So who are the amino acids that are responsible for the Fmax? You come back to the trajectory and you look at your, let's say, your system at Fmax, and then you see that here there are some amino acids that are pretty much the aromatic ones that are keeping the highest forces on the complex. So if you want to, so this shows that mechanostability on the protein complex, in this particular on the side of the nanobody, is very important. For the case of the wild type, fine. They engineered something that was very good for wild type, but it got worsened by the different appearance of variants, right? So that's the story. So if you want to engineer something that is more robust across different variants, not only consider the, let's say, the binding strength, that is very good, because that's part of the recognition, but once mechanical forces are, let's say, acting on the complex, so you will need to think about those amino acids that are lasting before the system separates, because those are the ones who take care of the, let's say, the stability of the complex. Right? So we went a little bit forward, and then we studied different variants, and also we looked at the different, let's say, correlations between the residues that are lying at the interface at Fmax, and we see for the same, let's say, nanobody, we see that this changes actually with the number of different, let's say, variants. XBB15 is one of the Omicrons, and so mechanostability is relevant if you compare these two. I mean, it's ceasing over the time, and this is very important to feature here. But of course, some amino acids, they remain stable, right? So those are the amino acids that actually try to retain the stability of the protein under mechanical forces, right? So if mechanical forces are acting at the cellular level, so we should also consider as a principle of design also the mechanostability of, let's say, therapeutic complexes. So we were actually, I started saying that we could study much more, and also was mentioning to Bogdan that we can study much larger systems. So yeah, so with the idea of enhancing the GōMartini 3 approach using the high frequency contacts. So here is, let's say, all atom studies, they typically now in modern days, they reach 100 microseconds, but these are limited to few cases. For instance, in the case of the SARS-CoV-2, it has been done by, you know, these Livermore and Oak Ridge national labs. Those are pretty large facilities where they, let's say, so they can do for one variant. They can run for hundreds of microseconds because it is computationally demanding to do those simulations, right? And if you think to scan in the space of different variants, so this will not be the solution, right? So you cannot use twice in a year this kind of gigantic facilities only for changing 2 amino acids at the interface of the protein, right? So our method has been tested. I mean, and this is the new enhanced model, so which basically we call high frequency contact. That is, I mean, if you look at the RMSD, the system remains stable over 1 Armstrong above the reference one. And if you look at the RMSD for this 108 global protein, so we have more also, you could see that the profiles are pretty much within the Hellinger measure, so they are below 0.3. So our model, the high frequency, is as good as the crystal one, so which means our system can be described with a new high frequency context. So if you look at now a different system, so now let's look at the spike protein. So over 100 microseconds of time scale. So now with different, let's say, across different variants of space, and you recreate all the pipelines of setting up GōMartini simulations. You could study this process, let's say here, I'm showing only 1 microsecond. So we have the atomistic, let's say the atomistic simulation also is around, let's say, this value. So we have pretty much, let's say, the system under control if we consider the same, let's say, level of uncertainty. And of course, in terms of the fluctuations, I mean, we can see that the secondary part of the protein, or the most relevant domains, they remain under the stability, as well as the, I mean, we compare also the using the Hellinger measure, so we can compare the distributions of, well, let's say, the optimized models. So we can see that they are way below 0.3 compared to the all-atom simulation. So that validates our approach. So our and then, so we went to scan, let's say, over those 100 microseconds to see more, right? All-atom simulations for the wild type stays in this blue square. So this is starting from the up conformation. All-atom simulation is able only to sample, let's say, this space in the, let's say, in this theta and d, which is basically the, if you consider the red protein, which is the RBD. So we measure this distance and the angular position respect to the axis. So the simulation stays in this area. So this is a coarse-grain profile. So for the wild type, so we see, I mean, we enhance the space by having, let's say, extended open transitions, open states, as well as transitions to the down state. You compare that with the delta, so then you will see that, that there are much more details depending on the position of the delta. Delta has about 10 mutations, and the GN1 30 mutations along the spike protein. So the profiles are very different, but you could see that the delta has been, I mean, has some states, open state, which is much more in the open, so which gives the ability to stay in the, let's say, in the active state much longer than any of the other variants. And of course, this is a hypothesis to say that delta was more pathogenic than any of the other variants that came later, right? And okay, so this is just I mean, I want to play this video. Going a little bit slower. Wait a minute, I know what to do. Okay, so these are basically the wild type, and this is delta. So basically, you could see the RPD, the up conformation stay in the wild type, while in the delta, you see with the GōMartini simulations, you see, well, basically this is the down state, right? So we see that transition much more often, I mean, which is not possible by the wild type variant. So also these are all atom simulations. So basically it's done over, let's say, you use 1.5 microseconds, but basically the whole system remains in that state, yeah. And the performance is also, you could see that this is 0.01 microseconds a day. That's what all atom simulations can do these days, even if you use a Hopper or any other facility, any other GPU graphics. So you won't go much more higher than 0.01 microsecond a day. While if you use our model, so you can generate 2 microseconds a day. Yeah. So this is much more realistic if you want to study those large conformational changes for the cases of protein complexes or protein assemblies. Okay, so basically, so we have I shown basically this model described with GōMartini. So we have better efficiency to sample the conformational states of Delta variant, and of course, I mean our work continues because we want to integrate also the other part, the anchoring part on the membrane, and we know that the spike protein also, we have all atom data for that, basically has different, let's say, conformations, which are pretty much enhancing, let's say, the dynamics of the full SARS-CoV-2, let's say, spike, yeah. And so this is a work in progress, but this shows already that we can efficiently capture, let's say, the different conformations of the opening and close for the case of Delta, and yes. Oops. This is just molecular dynamics. So you have room temperature, and then you perform the dynamics of the protein. So one basically stays at room temperature in the open state, yeah, while the other has some, let's say, space or some flexibility to transition from open to close. Some people also has shown also that this transition happens in the presence of some glycans, but we see that this process is not just triggered by glycan, but it's also intrinsic from the RBD, from the dynamics of the protein. Oops. Okay. So just to conclude, so I showed that the GōMartini 3 is a method developed over the, let's say, several years. I mean, the community is not seated here, but it's a lot of people also helping to not only parameterize, but to integrate the same ideas into the Martinized 2 platform, which is a more versatile tool that can be used by any person in GROMACS. Protein complexes, at the moment, they require the presence of co-interactions at the interface of proteins in order to describe not only the dynamics, but also the nanomechanics of those systems. And so the last part, I showed that the enhanced GōMartini 3 framework can be used also to study large assemblies of, protein assemblies. And of course, I mean, our dream is to follow up on the dynamics of these entire large systems and see them in action, right? And so the team, so basically, thank you for, everybody who has supported these ideas, and also, the lead of the Martini 3 community, the Martini Protein Task Force that is actually listed here. There are much more people joining this effort, and also the people who has also support us with experimental data in Poland and abroad also, and also my team who have been very supportive with these ideas and the implementations and the funding, and also former members of our team, right? And that's all. Thank you for your attention.
I will have questions. In all atom simulations, as I counted, they used, I think, 200 days to accomplish simulations.
Half a year, I think, for those simulations. It can be half a year of having the queue to put one system on the HPC and then let it run until generates all this data.
And you obtain the same results just for
weeks.
1 week.
Weeks.
Oh,
I mean, I think 10 microseconds we do in a week, so If we consider this as a replica, we can have 100 times those processes. In a week, we can reach 100 microseconds of cumulative MD simulation. Right?
And the results were quite similar, yes.
As we benchmark with their data. So if they have the wild type, so that's our reference data. So we try to benchmark our simulation with that, but then we explore different cases. That's where they don't have simulations, and that's where we gain insight from coarse-grained simulations.
Thank you. Please, Professor.
This simulation is based on atomic potentials, but what you illustrate, you have classified as this is tension, compression, and shear. How can you translate this continuum observation of compressive mode? You compress, you pull, you provide some failure, like cracks. But as regards shear, you should represent some fundamental properties of such mode of deformation of such system. But you didn't discuss it. You mentioned that shear stress in some pictures, but you didn't present fundamental properties. How the shearing progresses in such
We have this. Yes, this is a valid question. So from the mechanics, I think from the continuum level of theory, I think there is much more broad definition of the shear definition. In case of these are very molecular systems, I believe the definition is quite faulty if we consider smaller systems like small proteins. But if we increase the size of the system, then we can increase much more, let's say, to filaments of proteins. So then we could understand a bit better, let's say, what does it mean shear propagation or force propagation or crack propagation in the, let's say, in the protein space, which has to be related in this case to the sequence of the protein, right? So we can imagine that this will happen once we increase the size of the system. At the moment, we are still at the length scale that we have, let's say, hundreds of. We are not even yet at the micrometer length scale. We are at the nanometer, tens of nanometers. This is more or less the size of the spike protein, 30 nanometers. And of course, I mean, if we have much more protein assembly, larger systems, I guess the definition of what you call shear definition can be also inside these systems. At least for the polymers, people use shear definition, polymer melts. They use the same definitions. So we could imagine that this definition will hold also from biomolecular systems, but we don't, I don't know that yet.
You increase about metals. You have monocrystal, then you have very clear understanding what is shearing, what is slip weight, and patterns like shearing. But in such system, you could probably should study fundamental how behave such system under shearing. Whether there are some similarities to monocrystals.
Could be. Yeah. No, I understand. We can do. I think this method is that. I mean, if we forget that these are proteins and they have a sequence, and this sequence has different pattern, and they make the bulk. If we consider this as a bulk, then perhaps we can trace, let's say, the profile of the forces going through the bulk. This is something that can be traced in the simulation. I mean, how the forces are being from the active side, where we are basically loading, adding some forces, how the force propagates through the system, because this is in the end how it happens. MD simulations know how the forces have been transferred through the different amino acids up to the end of the protein complex surface, and then goes to the other protein. But we need to check that carefully. I think we have methods to check the pattern of the force through the protein complex, but we haven't checked that.
Maybe Professor Postek, because you are working in molecular dynamics. Oh, please, Professor Paszek.
Adolfo, you know, I was looking at your movie of the changing conformation. I was wondering about biological interpretation of this, right? So this is autonomous system, right? There is no change of condition, the one variant, right? Changes between the two states. What does it mean biologically, right? There is a time scale. This is is this a steady state? Is this some sort of stochastic equilibrium when protein is changing between those two conformations? How often does it happen?
Yeah, those are all good questions. And we have the free energy landscape, so we can actually run some Markov state model to check on the dynamics of these transitions, how often they happen. So, but we haven't done yet. This is also possible, but more in the biological. I think the interesting thing is that if you consider the spike protein with the RBD in the open conformation, that's unstable. It's in non-equilibrium, right? That RBD is looking in the let's say in the case, it's looking for the AC2 for the receptor to bind and lower the free energy. Once this happens, it will just stick to the cell, and then it will be the lower in energy. Once it is in the open state, it's basically transitioning between open and closed. And this process is being enhanced by the presence of mutations. That's the only thing. So delta has, let's say, there are some studies, yes, some experimental data where they check the population of open and closed state for the wild type. So it used to be 50-50.
Is the open state required for binding, or what's the function?
Without any cell. So without cells, so the spike remains. Once they check this with some method, so they can see that there is a 50-50 chance that the spike stays in the up or down conformation. And computationally, it was shown that the open state is being triggered because some glycans kick the RBD to the open conformation. And what we show is that, of course, if you take all atom simulations and some time scale, so this RBD remains in the open state. This is what happens in the all atom case. But if you use a different variant and then you move, let's say with a let's say in longer time scales, you will see that this process happens much more often. And actually, it stays in the open state much longer time than the down the state. So this, but of course, I mean, we can check the kinetics of the process, and if we have more data also, we can see those rates also that are very interesting.
Okay, so my question is whether in all atom simulations do we take into account quantum mechanics or not?
No, but actually that's a good question, because the machine learning methods that I highlighted at the beginning, so there are some of them, especially the ones that are still slow in the, I mean, like we are also mentioned, but we are sort of studying also that. So these, well, these are coarse-grained methods, of course. I mean, this is, here it starts. Solar is one of those machine learning methods. I mean, these methods are concurrent to all atom simulation. So all atom simulation doesn't take care of anything from the quantum world. Right? So they are classical models, but the machine learning methods like this one, like that maze, like Annie, so those are methods that actually are being parameterized to reproduce also quantum information at the classical level. So you have, let's
quantum
say,
physics.
Yeah.
What does it mean, quantum?
Quantum,
information?
yeah. What quantum information? Let's say if you take the energy of binding which can be purely quantum. I mean, if you take some, let's say, some systems that are not well described because they have some degree of polarization. So classical models are not able to describe that quantum, and because it's a quantum effect, and that if you parameterize that in machine learning, so then you can gain information about these binding energies better than in all atom simulations. So yeah, we are still in the level of classical models, let's say, and okay, so we expect that quantum effects are not when the systems become very large also.
Very large in the context of many particles, yes?
Many particles, micrometer length, microsecond time scale.
Okay. Maybe a very naive question at the end. So I'm wondering, because these systems are very, very complicated and so on, and some potentials are shielded and so on. So how do you think, where from the success of these models by introducing Lennard-Jones potential? So these exponents 12 and 6. It's really...
Why so magic that 12.6 Lennard-Jones? I mean, I mean, you could parameterize anything, right? So I think the Go model was parameterized with different rules of 12.6, 10.4, and Yukawa, and some other potentials, but the one that performed the best was the Lennard-Jones.
But and you only use 12.6, yes?
12.6, okay. yes. And also the Martini is also 12.6. If there are ideas also inside the community that we should leave the 12.6 potential, that the system will be, and there are cases in which 12.6 is too restricted because, I mean, you have this full repulsion. It would be too heavy for some cases of some constructs of biomolecules. RNA, I think it must be, they think that it should be softer detail, and so, but at the moment it's a matter of choice, right? So we have the 12.6 as a technical level, ground floor for any implementation.
Okay, and for me it's very intriguing. So if there are no other questions, let's thank the speaker again.
Thank you. Okay. Yes, okay. At least one person. Okay. Tomorrow I will have a meeting with them. Everything was. I will ask the lady how was it. I just think that we can get to very large systems.
Well, conditions are continuous.